All right, everyone. Today we will discuss cell cycle and mitosis. I have combined the content from two different chapters for you to have a better understanding. So the page numbers are listed for your convenience and also the chapter numbers here too. We have learned that DNA can exist as two forms, chromatin and chromosomes. And we also learned that a chromosome is a more condensed form of a chromatin. The chromosomes have this X-shaped structure, and there are a few structure names on chromosomes that I would like you to remember. First is the chromatid. Okay, as a chromosome replicates or makes a copy of itself, it's arranged as two chromosomes next to each other, called chromatids. Okay. Second is a telomere, right? So telomere is a region at each end of the chromosome and protects the end of the chromosome from deterioration or from fusion with neighboring chromosomes. Okay. The third one is called a centromere. So centromere is the region that joins the two sister chromatids or each half of the chromosomes. During mitosis, the spindle fibers attach to the centromere, all right? So I'm asking you a question. So when does chromatin become a chromosome? So the chromatin contains unwounded DNA and is found in cells that are not dividing. In contrast, chromosomes are tightly packed DNA found only in dividing cells. Shown in this picture, those are dividing cells. In some of the cells, you can clearly see those secant condensed form of chromosomes. There are two types of cell division. One is called mitosis, shown here, and the other one is meiosis. You should have learned this in cell biology or genetics before, but I would like to emphasize the differences between these two types of division. Okay, mitosis is the asexual division to produce more cells. This is for the purpose of cell growth or tissue repair. We know that human cells have 23 pairs and 46 chromosomes. And having two sets of chromosomes, we name it as a diploid, shown as 2N in these slides. During mitosis, the cell with 2N produces two identical 2N cells, and only one division happened during this process, shown by one arrow. In contrast, meiosis is only involved in sexual reproduction, meaning the meiosis only occurs in the process of generating sperm and egg. Another difference is that the meiosis has two rounds of division, shows as two arrows here. Therefore, 4 germ cells will be produced at the end, and each of them only has one set of chromosomes. So we name it as haploid, shown as 1N, okay, or N. So when the egg is fertilized, the genetic material of the egg and the sperm, they will combine and become 2N again. So this is a diploid cell. To prepare for cell division, the cell will go through a series of events to accumulate enough materials and get ready for division. Scientists named the whole process as a cell cycle. There are 4 main phases in one cell cycle: G1, S, G2, and M, which stands for GAP1, synthesis as S, GAP2, and mitosis. If the cell is not in cell cycle, meaning the cell is not actively dividing, we consider the cell is in G0 phase. Okay, this is shown here. It's not included in the active cell cycle for G0 phase. But when people grow to an adult, most cells in our body do not divide anymore. So which means that most of our cells stay in G0 phase. But during the embryonic development, cells are actively dividing. And you can find cells in all different cell cycle phases. But what kind of events are happening in different phases of cell cycle? This slide lists a few, okay? So as we mentioned, G0 basically have no cell division, therefore no DNA replication is necessary. G1 is the longest phase, and the cells make more organelles and increase in cell size. S phase means DNA synthesis phase. You can imagine that genomic DNA is synthesized or replicated during this phase, right? So only the whole set of DNA is completely duplicated, right? And then once S phase ends and the cell enters the second gap phase, that's called a G2 phase, okay? G2 phase is relatively short. And during this time, the cell continues to make organelles and increase in size and get everything ready for this upcoming mitosis. So mitosis is the shortest period in the whole cell cycle. However, because scientists can clearly see chromosomes in mitosis based on the shapes or the changes in cell, the mitosis can be further divided into 4 subphases, including the prophase, metaphase, anaphase, and the telophase. Okay, we will talk about them in detail in the next few slides. Let's first watch a video about cell cycle.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
I prepared this slide for you to remember the 4 phases of mitosis easily. This part is frequently tested in MCAT exams. Remember the name as PMAT, which stands for prophase, metaphase, anaphase, and telophase. Also, prophase is when the cell prepare for the mitosis. So it prepare also starts with a P. And metaphase is where you can see the chromosomes align in the middle of the cell. So it start with an M. And then anaphase is the sister chromatids are pulled apart. So you can see the apart start with A as well. And the telophase, you can see the shapes of the two daughter cells, which two daughter cells start with a T. So then you can easily remember the different phase and what they look like on the picture. The video we talked about cytokinesis, which ends the mitosis. There are two views of cytokinesis on this slide. So A shows a scanning an electron microscope view of the cytokinesis from the outside of the cell. And the B shows a transmission in electron micrograph of cytokinesis in the cross-section of a dividing cell. Remember that not only the chromosomes, but also the organelles are distributed to the daughter cells. So the dark substances shown here are the chromosomes. And then over here, you can see those are probably mitochondria, also separated into two daughter cells. Now let's get back to a picture I showed you before. So can you tell which mitotic stage are these four cells, 1, 2, 3, 4? You can pause the video now to think about it. Okay, yes, these are the P, M, A, T respectively. That's prophase, metaphase, anaphase, and telophase here. So if you did not get this correct, please make sure that you go back a couple of slides and try to remember this again, because this is important for the future exams. Okay, so the question here is, are there ways to detect the cell cycle phases other than using a microscope? Here, I would like to show you a popular technique to identify cell cycle phases using a technique called flow cytometry. Okay, so this is a equipment named flow cytometer. They need to be used for detecting cell cycle. And the machine actually is be able to make measurement of the cells in solution as they quickly pass this instrument's laser. See, this is inside the instrument. They have a laser here, and the laser is going to point it to every single cells that passes through this little tubing. Okay. And then if the cells are labeled with fluorescent dye, the fluorescent signals can be detected and quantified by the machine. It is very common to use flow cytometer to analyze cell cycle. All right, so when this experiment is performed, the cell DNA is stained with fluorescent dye. Therefore, as long as one cell passes the laser, the machine gives a count, gives count. So a typical result is shown here on the right. So usually two peaks are observed, one peak and second peak, okay? The first peak is usually the cells in G1 phase, and the second peak is usually cells in G2 or M phase. And cells in S phase are accumulated in the area between this G1 and G2M phase. So why this is the pattern? Okay, so I hear you show, first of all, this is G1, G2, or M, and this is S phase, all right? So remember the value in x-axis, this is called DNA content, right? Because we labeled it with fluorescent dye. And the y-axis is the numbers of the cells being counted. We just learned in a normal cell, right? The DNA content is 2N. So the DNA content in G1 phase should be 2N because this is just normal cells in the G1, the longest, the normal phase, right? However, in S phase, DNA is gradually synthesized, right? So the DNA replication occurs in S phase. And at the S phase, DNA gradually synthesized, then until the full copy of genomic DNA is replicated. Therefore, you can imagine the S phase can be anywhere between the 2N to 4N content of the DNA. But at the end of the S phase, you can imagine the full copy of genomic DNA is replicated. Therefore, by the end of the S phase, the DNA content in the cell is supposed to be 4N. And then at the G2 phase, the cell is going to keep make this 4N content there until N phase. The cell divide from 4N into two daughter cells. That's each of them contains 2N. All right. So remember, the 4N peaks always double the 2N. That means here you're showing the 2N at G1 phase. The highest peak is around probably 75, maybe 70, I would say. And then G2N phase, the peak is about 140. So the number is doubled. All right. So imagine if it's 8N, how much is going to be there? So 140 double again, then it's going to be 280, which is already past this chart. Okay. So now let's think about the question. Why G1 phase have the highest account here, the highest peak? You can pause here. So the answer is most cells spend most time in the longest time in G1 phase. All right. So similarly, in which phase the cells have the lowest account? Yes, M phase, because the M phase actually is the shortest time. The longer you stay in a certain time, that means during this time, there will be more cells staying there. Therefore, the peak is the highest, right? If you have a shortest time, then you have a shorter, the less cells will accumulate during that particular time. Therefore, the number will also low. So if like a cancer cell line has a doubling time, that means doubling time usually from you double the number of the cells, that called doubling time. So the cancer cell line, if average is probably 24 hours of doubling time, then the M phase may only last about 1 hour. Okay, so G1, the time probably lasts for half of the cell cycle. That means probably 12 hours. That's an estimation. So that's the reason why you observe cells under the microscope. You probably can find only a few cells in mitosis. Okay, but this peak actually contains both G2 and M phases, so they calculated together. Now let's take a look at a real example. All right, so I randomly choose one picture from a publication in 2013, which shows a typical cell cycle analysis experiment. So the top picture here is a control sample that shows a normal cell cycle pattern of a cell line used in this paper. Okay, so now you should be able to identify different cell cycle phases. So the G1 phase, G0 non-divided cells also have 2N. So in this paper, to just label them G0 and G1 together, okay? So they're using a software named FlowDraw for this analysis. And the FlowDraw software can quantify the percentage of the cells in this picture. For instance, here they show this 46, 33, and 21 that indicate the G1 or G0 phase has 46% of the cells, S phase has 33% of the cells, and G2M phase, they have 21% of the cells, okay? The figure in the middle, this one is a cell cycle analysis. And then showing here, there's a huge peak on the G2M. And then this distribution percentile is G0, G1 is 2, and then this is 8 and this is 90% in the G2M phase. But then here they label the R0, RO3306 and 10 micromolar 24 hours. That means is the cells were treated with this compound, means RO compound, for 24 hours at a concentration of 10 micromolar, okay? So you can tell the pattern is a huge change between this top figure and the middle figure, right? So this suggests the cells, once they go process to the G2M phase, they are going to stop here and cannot continue to go to the G1 phase anymore. So we call this pattern as G2M phase arrest, okay? In the bottom figure, so basically the authors remove the compound and change to a regular culture medium for 1 hour. So this is 1 hour. WO means without. So without the compound, they culture the cells for 1 hour, okay? So what will happen? So you can see here that after 1 hour of culture, we remove this compound, there's a small G1 peak starting to show up, right? So indicating that the effect of this RO compound on G2M phase arrest is likely reversible because they release some of the cell from here and then they process into the G1 phase. Okay, so make sure that you understand these figures in order to make correct selection in the future exams, all right? So if you are curious enough, I think now you may ask, why does cells show cell cycle regulation? What are the driving force of the cell cycle? So when you look into the biochemical molecules involved in this process, we call this as molecular mechanisms. Actually, cell cycle is controlled by a group of proteins named cyclins, okay, all these different cyclins. So this figure shows our 4 major cycling proteins. Their concentrations in cells that changing in different phase of cell cycle, okay. For instance, this cyclin E also is a G1S cycling, express a high in G1 or S phase, this transition phase, but cyclin D1 instead will express very well through almost all those whole cell cycle, except for, I think, after mitosis will decrease a little bit. So the changes of those cell activities we observed the combinatory effect of all these cell cyclings, okay? So then again, if you are even more curious, you may ask, what exactly these cyclins do when they are expressed? Why this level is correlated with different phase of cell cycle? So in order to answer that question, we need to introduce another group of proteins named cyclin-dependent kinases, or in short as CDKs. What do kinases do? You already learned that, right? Kinase phosphorate proteins. We name the proteins being phosphorylated as substrates. So CDKs are enzymes that can phosphorate substrate proteins. By doing so, CDKs become the driving force for cell cycle. However, the CDKs do not have kinase activity by themselves. Okay, when they are alone, they don't have any phosphorylation ability. Okay, and they need to bind to cyclins to become active. That makes the cyclins very important. Also, that means why these names are called cyclin-dependent kinases. It has to bind to cyclin first in order to become active. Okay, the students who would like to go to graduate school would probably will ask, how does this exactly happen? All right, this slide will give you some ideas. On the left, without cycling, the CDK does not have activity alone, all right, because its current structure is not appropriate for it to have the kinase activity. But on the right side, okay, when in certain stage, the cell start to make cycling. We know the cycling is like changing its expression along with the different phases, right? And then here shows a G1S cycling, such as cyclin E maybe. Okay, the binding of the cyclins to CDKs change the structure of CDK, now activating its kinase activity. And as they working as a complex, CDK can phosphorate its substrates like target 1, 2, 3 phosphorate shown here. And these substrates now can function to drive the cell cycle into the S phase entry, okay? So that's how S phase begins. Here, since we discussed about kinase and phosphorylation, and some of you may know or learned from other courses that phosphorylation is the key event to drive cell signaling pathways. But why phosphorylation is so important? The kinases add a phosphate group to specific amino acids such as serine, threonine, and tyrosine. Remember that phosphate group shown here is a negative charge, right? So if the charge of the amino acid changes, there is a good chance that the structure of the whole protein will be changed. If you learned about chemistry, you will understand that the function of the protein is determined by its structure, right? So this is the reason why phosphorylation can change the function or activity of a protein. Now, because the concentration of cyclins alters during different cell cycle phases, you can imagine the activity of CDKs also change accordingly. So even though their proteins probably remain constant. If we consider cyclins and CDKs as engines for cell cycle, how about brakes? You cannot just have engine but without brake, right? Similar to cars, our cells are smart enough to have a brake system. We name them as like checkpoints. Okay, that's what we are going to talk here. During checkpoints, the cells can examine if they are ready to go to the next cell cycle phase or not. If the error comes up, the cells will hold the cell cycle and try to fix the errors. For holding the cell cycle, we call the cell cycle arrest. We just talked about the G2M phase arrest as an example previously, right? So those indicate the cells have detected something wrong during the G2M phase, so they can have a rest. We will discuss about DNA repair later, but here let's first see what kind of checkpoints can happen, okay? Listed here are 3 major checkpoints. You can see these checkpoints are usually occur right before the cell enters next phases, all right? This include G1, G2, and M phase, those checkpoints. It is extremely important to keep your genome DNA secure and accurate, and cell cycle checkpoints are one major safety mechanism. So otherwise, after many rounds of cell division, there will be too many mutations in the cells. That's not going to be good for us, right? So now let me ask you another related question. Can you think about another ways to protect our genomic DNA as a safety mechanism? Right, we already talked about the nucleus. All of our genomic DNA is limited, contained inside the nucleus, not in cytoplasm. So this is actually another way to keep the integrity of our genome, okay? Now I will use G1 checkpoint as an example to describe how our cells do this. For instance, if during the G1 phase, our genomic DNA somehow get damaged, as the picture shows, what will happen at the molecular level? Interestingly, our cells will sense this DNA damage and trigger the expression of a protein named P53. P53 is one of the most important and famous protein in cancer biology I would like to remember you guys to remember the name of P53. The official gene name is TP53. Okay, P53 is a transcription factor. I told you that function of a transcription factor is to activate a gene transcription. Remember that? Okay, P53 is a transcription factor that can induce a group of genes. Okay, and then some of them is also CDK inhibitors shown here. As the name indicates, CDK inhibitors can interact with cyclins and the CDKs. All right, it can inhibit the kinase activity of CDK. So basically, the brake is turned on and stop the engine by the CDK inhibitor in this way. Okay, so then the cell cycle is held and the cells are paused in G1 phase, meaning G1 phase arrest. All right, so what the cells do during arrest? In this particular case, the cell will try to fix the damaged DNA. Okay, once this damage is fixed, the cell cycle will resume. However, if the cells has a very severe DNA damage and cannot be fixed anymore, what will happen? Then the cells will go apoptosis. That's a form of programmed cell death. Okay, you may ask then, what does this have anything to do with us? DNA damage actually happens every second in our body, no matter what you are eating, no matter where you are studying, sleeping, or even playing games. I'm giving you a number. So naturally occurring oxidative DNA damage arrays at least 10,000 times per cell per day in human. Okay, so we cannot prevent this from happening. We have one lecture designated for DNA damage, and I will not get into those details for today, okay? So cell cycle is tightly linked to cancer. Cancer can also be considered as a genetic disease, and some scientists think that cancer is actually a disease of the cell cycle. There are reasons for that. When you are young, so our cars are like a good car, our body, our cells like a good car, so they know when to start the engine and when to stop. However, when people get older, like me, more and more mutations are accumulating in our cells. And some of these mutations could lose control on the engine or the brakes, okay, and leading to an uncontrollable cell proliferation. And in this case, tumor can form as a result. Okay. For instance, the P53 protein we just discussed has found to be mutated in more than half of all cancer cases, indicating that a proper control of cell cycle is extremely important to prevent cancer development. We will discuss more examples like P53 in just a few slides. Let's take a further look on how cancer is developed. So you can imagine when even one cell loses control of a cell cycle, it can become 2, 4, 8, or become tumor in the future. So therefore, cancer cells can be clonal descendants from one mutant cell. Also, it is actually not super easy for a cell to become cancerous because the specific mutations can take time to build up, right? So this is the reason why cancers tend to be more prevalent in old people, not in young adults. The early onset of cancers usually associated with germline mutations, which we will talk in a few minutes. Here, I would like to emphasize that cancer usually can cause patient death if it's localized. Benign tumors are the examples. It can be really big, growing in one side, but not, cause the patient's death. However, metastatic tumors, meaning the cancer cells are invasive and can infiltrate surrounding tissues, that can be very dangerous. This picture shows what metastasis look like. Okay, if the cancer cells develop in the local site and it's not relocated to anywhere else in the body, we call them primary tumors. But once the tumor cells accumulate enough mutations, giving them ability to migrate, these cancer cells can move to new locations in the body. Usually this process is through the blood vessels or lymph vessels. That is the reason for certain cancer diagnosis, a blood test is to identify the cancer cell markers. These markers are special to cancer cells only, okay? If the cancer markers can be detected in blood, this will indicate that cancer cells have entered the peripheral blood. So meaning the cancer is metastasis, okay? Another evaluation is looking to lymph nodes. If a surgery is done to remove the tumor, several lymph nodes close to the tumor site are usually taken for histological examination. The pathologist can tell if these lymph nodes contain any tumor cells. A positive result also suggests that the tumor is metastatic. The metastatic tumor is much more difficult to cure than non-metastatic tumor. Generally speaking, based on the origin of the tumors, cancer can be classified into two categories: sporadic cancer or inherited cancer. Most cancers are sporadic, meaning the cancer is caused by gradual accumulation of mutations in key genes, such as p53, as we mentioned previously. In this case, most likely only the cancer cells carry the mutations, but not all the other cells in the body, okay? But in contrast to that, cancers can be inherited as a germline mutation. That means the mutation is inherited from one parent. The inherited cancer cases are much less, only accounts for less than 5% of all cancers. One major mechanism of inherited cancer called loss of heterozygosity, okay, also called LOH. So I'll give you an example to explain. This is this in a few minutes, okay. Let's first discuss of the sporadic cancer. This is very straightforward. When a cells divide, mutations can occur. This could be due to the error in DNA replication, oxidative stress, or simply just got too much Texas sunshine in the noontime in summers, right? So over time, cells acquire more than one mutations. If the cells is not lucky, maybe that was the reason why they are in blue showing here. And then two independent acquired mutation in the same cell may be sufficient to cause uncontrolled cell growth and cancer. You can tell that those unmutated cells, those cells are okay. And with one single mutation, which is controlled still, they are okay too. Scientists have classified mutations on cell cycle related genes. You may have noticed sometimes I say gene mutation and sometimes I say mutated proteins, right? So remember that the mutation indeed first happened at the genomic DNA level, and these DNA mutations may cause mutated proteins, right? So we have one question in our sample test asking whether DNA mutation always generates a mutated protein. When this mutation is a silent mutation, this amino acid sequence of the protein will not change. In future lectures, we will discuss that in detail. Here the mutations can be classified into two categories. Tumor suppressor genes that works as brakes. One example is p53 gene. Okay, we will also talk about another famous tumor suppressor gene named RB1. Another class is called a proto-oncogene. So these genes have an opposite function of tumor suppressor genes. proto-oncogenes drive the cell cycle and promote cell division. Okay, we all have proto-oncogenes in our body, and they are responsible for normal growth of our cells. Normally, they are under control, and they are turned on only when necessary. However, certain mutations on these proto-oncogenes can turn them on continuously, meaning they become always active, okay? Under this condition, we call them as oncogenes. So I bet you definitely heard of this term. I'll present an example of a proto-oncogene named RAS. So the difference between proto-oncogene and oncogene is important. Please make sure you understand it, okay? Here, this slide quickly shows you that in A, shows a protein encoded by this normal tumor suppressor genes. They can inhibit cell division. This is the protein, inhibit cell division. And then if the proteins encoded by a mutated tumor suppressor gene, they can fail to inhibit the cell division. And then the cells are uncontrolled to divide into more and more cells. Here is the tumor suppressor gene named RB1, we just mentioned. So the name of RB1 came from a pediatric cancer called retinoblastoma. This is a pediatric malignancy that most commonly affects young children. The tumor begins in the retina and may occur in one or both eyes. Currently, the treatments include chemotherapy, radiation, and laser therapy. Mutations on RB1 gene can be sporadic or inherited, and this slide is focused on the inherited form of RB1 mutation. That means germline mutation, okay? Specifically, the development of retinoblastoma needs to go through the process of LOH, or loss of heterozygosity. It's not LOL, okay? The cells shown on left here contains 2 copies of chromosome 13. This 13 harbors the RB1 gene, and both alleles, they make RB protein from both alleles, okay? So we know that for everyone, we have one set of chromosomes from mom, another set from dad. So we have abundant this RB protein. However, in children who carries RB1 mutation, one of the 2 chromosome 13 has a defect on RB gene and fails to make RB protein, shown as the blue chromosome is a defective. Maybe it's, most of the cases, it has a deletion on the RB gene, okay? It does not make a RB protein, but they still maintain a normal allele that keep making RB protein, all right? But then this cell is at risk because only one allele is making this protein. Since the 2 alleles are different, we learned from genetics that this cell is called a heterozygous cell, right? Remember? So in case one cell have a mutation event that disrupt the normal allele, then in this case, no RB protein will be made, right? Because this no RB, we know that RB is a tumor suppressor gene. That means it has a chance to this lead to cancer development. So this is the process of loss of heterozygosity. The heterozygous status is no longer existing anymore. So this is LOH, okay? LOH is a hallmark of tumor suppressor genes. Like the P53, we'll talk about this, a tumor suppressor gene, is also have this loss of heterozygosity as well. Okay, so this slide shows how RB protein work as a tumor suppressor protein. Okay, when RB is active in the nucleus, it binds to this protein called E2F. So E2F is a transcription factor that controls expression of a large gene set encoding genes that moves the cells through the cell cycle, okay? So when bind to this RB protein, so E2F becomes inactive, and the cell does not move to G1 to S because it's inhibited, okay? And interesting, this RB protein can be phosphorylated, so become a phosphorylated form of RB. You can see once phosphorylated, they lose the ability to bind to E2F anymore, okay? So this will cause E2F to released and starting working as a transcription factor and going to transcribe a lot of cell cycle related genes. So if think about in the retinoblastoma cases, if the RB is missing or mutated, it does not have any RB in there, what happened? So E2F is always active and the cell grows and divides continuously, forming cancerous tumor. All right. So if the RB protein can be phosphorylated, then the kinase should be important for cell cycle control. Is that correct? Yes. So do you know which kinases phosphorylate RB in this event? We learned that they are actually CDK. CDK4 and 6, they can partner with cyclin D and phosphorylate RB. So then you may ask, how important this? Is studying this CDK or cyclin related to clinical application? I'm going to give you an example, okay? So let's first spend some time to see how breast cancer cells grow. We know that most of breast cancers happen in females. Indeed, the males can also have breast cancer, but very rare, okay? In females, the hormone estrogen is an important hormone for breast cancer to grow under the normal condition. Many of the mammalian gland epithelial cells contain this transcription factor named estrogen receptor, okay? So which has a high affinity and can bind to estrogen. Once the estrogen goes in, estrogen receptor, this protein can bind to estrogen. Upon this binding, the estrogen receptor becomes active. It's a transcription factor and can induce expression of many genes that are required for cell growth. All right, so then you can see this cell's active transcription and then promote cell growth and divide it to even 2 cells like here. And then in cancer cells, about 70% of the breast cancer cases are estrogen receptor positive, meaning those cells are use this mechanism to grow, all right? In order to slow down or even stop the cancer cell growth, the scientists have developed many drugs to target the estrogen receptor action. So overall, it is called a endocrine therapy. So it will block the estrogen receptor to transcribe its downstream genes. But after a certain time, the therapy will induce drug resistance and the cancer can come back, okay? The resistance can always happen. Tumor relapse is not a good sign and can take away patients' lives, all right? So interesting, a few years ago, scientists found that these endocrine resistant tumors, their expression level of CDK4 or 6 become extremely high, actually maybe hundreds or even thousands folds of higher. So basically the tumor cells trying to increase the protein level of CDKs to drive the cell proliferation, even without the existing of the estrogen receptor signaling pathway. And this finding led to the development of several CDK4 and CDK6 inhibitors. So structurally, these 2 kinases are very similar. So the inhibitors can inhibit both CDKs. So the name is drug abemaciclib and palbociclib were approved for clinical use by FDA in 2017. And ribociclib was approved 1 year later. They all have been used for treating metastatic breast cancers now. So, but then they are also very expensive, right? So the price for 30-day supply of each drug is at least $10,000. So you can see that the basic research gives idea for treatment, which not only can help the patients, but also can make big money for the pharma companies. So we learned all this cyclin D together with CDK4/6 phosphorate RB, and then RB phosphorylation lose the control of E2F, and then lose control of the cell cycle progression. So that's how it works, right? Okay. Okay. Oh, this last slides. We don't want to skip it. Okay. So let's talk about the proto-oncogene using RAS as an example. As I mentioned earlier, a proto-oncogene is a normal gene, shown here, and then its activity is controlled well when it's normal. The RAS proto-oncogene here encodes a signaling transduction protein that processes signals for cell division and transmit them from the cytoplasm to the nucleus. However, RAS gene can have mutations, here shows 2 sites, on 12 or 61, and cause the formation of an oncogene that permanently signals for cell division. So scientifically, we call this phenomenon as constitutively active form of RAS, okay? Many oncogenes are cell cycle regulated genes. So mutated RAS protein change conformation, remains active, and then they will promote tumor formation, right? So that's all for today. I'll see you next time.